Hello and welcome to your online lecture for the course Altered Physiology 1. I am Dr. Patricia Riley and in today's lecture we are covering neurologic disorders. This is the second lecture for neurological disorders. We'll begin and spend most of our lecture talking about common neurodegenerative conditions and diseases, and then we will finish off by discussing some of the common neurodegenerative disorders that affect children. So let's first discuss Alzheimer's disease. As you can see here, there are three different types, with the most common being the late onset Alzheimer's disease. It is the most common because when we look at the age of onset down here, you can see that 50% of people over the age of 85 have Alzheimer's disease. There's also early onset familial and early onset Alzheimer's disease, which is very rare. Now, when we look at the diagram on the right, specifically here, we can see that in a normal, healthy older adult, there is a normal brain weight reduction by 15% or of 15%. So that is an older, healthy adult will have 85% of the brain weight that they used to have. Atrophy of the brain is a very common function of age. The changes that occur in Alzheimer's disease include a decrease in acetylcholine in the brain, which leads to shrinking of the cerebral cortex, or what we would call accelerated cortical atrophy. So brain weight loss is greater in people with Alzheimer's disease when you compare to that of normal aging. In addition to the cerebral cortex being affected, we also have the hippocampus that is impacted. These areas are mainly involved in memory, thinking, and overall cognitive function. So if we look at some of the effects of Alzheimer's disease, we will see that cognitive dysfunction involving memory loss is seen, issues or loss of higher order thinking. Higher order thinking is where you're going beyond basic levels of thinking. And also issues with focus. So where are we here? Issues with focusing. And disorientation, so loss of sense of direction, really a state of mental confusion. There's also behavioral changes that are common, such as irritation and agitation, and mood swings are also commonly seen. And these symptoms that I just discussed are symptoms of dementia. And Alzheimer's disease is the leading cause of neurodegenerative dementia. Once other causes of dementia are ruled out, then the diagnosis of Alzheimer's can be made. So when people present with these symptoms here, it's important to rule out other potential causes of dementia before making the diagnosis of Alzheimer's disease. Okay. Next, we will move on to some of the pathological changes that are seen, or pathological alterations that are seen in people that have Alzheimer's disease. So this slide gives you an overview of these changes. On the far right, you can see a healthy brain versus one that is suffering from Alzheimer's disease. You can see in the healthy brain, it's very plump and full. The sulcus grooves are not very deep. But in Alzheimer's, you can see much larger gaps present. The grooves are much deeper. There's much deeper sulci in the area of, in particular, language and memory. And this is due, of course, to atrophy. And what we end up with are plaque formation. And these plaques are made by a toxic amyloid beta protein that ends up accumulating, and when it accumulates, it ends up forming plaques. And what plaques do is they disrupt the normal nerve transmission. These plaques disrupt the communication between nerves, and then the nerves can end up dying. So that's one of the causes of Alzheimer's disease and the changes, the alterations that are seen that are pathological in Alzheimer's disease. The second cause is neurofibrillary tangles. And in the diagram over here, you can see a nice clear cell body in a normal brain, a normal healthy neuron. But in Alzheimer's, you can see, if you were to zoom in, you would see that it's a lot more messy. It's tangled up. It's not a nice clear cell body. And you can see the tangling here, and you can see the tangling down here. We call those neurofibrillary tangles. Now, if you look at the diagram of the nerve here, you can see these green structures that are running vertically, and these are called microtubules. And microtubules are responsible for transporting vesicles that contain neurotransmitters down the length of the axon. And this is obviously important for normal communication and normal healthy neurons. But in Alzheimer's disease, these microtubules end up becoming detached. And they become detached because of the presence of tau protein. And tau protein is going to cause these microtubules to detach and form tangles. And when these form tangles, as you can imagine, it's going to prevent the proper transport of these vesicles that contain neurotransmitters. So not being able to transport these vesicles along the course of the nerve means that we're not going to get that release of the neurotransmitters once we get to the axon terminal in order to communicate with the next receiving neuron. So what this does is it ends up contributing to neuronal cell death. And you can see that there are two pathways that contribute to the death of the neuron. So those two pathways being that of the formation of these neuritic plaques and neurofibrillary tangles. And most of the neuron loss are those that would contain acetylcholine. And these are concentrated in areas of the cerebral cortex and the hippocampus, as mentioned earlier, which we know are areas that are involved in cognitive abilities and memory. And that's why we see deficits in these areas with these patients. So loss of the cholinergic neurons due to degeneration of the basal forebrain with acetylcholine loss. And then, of course, the location of plaques and tangles, the more concentrated they are in the cerebral cortex and the hippocampus, the more degeneration we'll see. So let's move on now to another neurodegenerative disorder called Parkinson's disease. And the 5 causes include idiopathic, so unknown, post-traumatic, toxic, genetic, and viral. Age of onset is usually earlier than what we would see in Alzheimer's disease, being between the ages of 40 and 60. It is more prevalent in males. The typical stooped posture in this picture here of someone with Parkinson's. And let's look above here at a particular area of the brain called the substantia nigra. And the substantia nigra gets its name from the black sort of dots that you would see that are present within this area that are made of melanin, which is used to make the neurotransmitter dopamine. The cell bodies that make dopamine terminate in this region here. So in the putamen, the caudate nucleus, and the globus pallidus. So the cell bodies are going to where we make dopamine are going to terminate in this area here. And collectively, this is oftentimes referred to as the basal ganglia. So in Parkinson's, the dopamine-containing neurons are lost or they die off, and so less dopamine is produced and therefore released in this area. Some of the main effects that are seen with this disease include abnormal movements, such as Parkinsonism, hypertonia, which is tremors, rigidity, hypokinesia, a flexed, shuffling gait, or slurred speech. If you haven't heard of the term Parkinsonism before, it's any condition that causes a combination of the movement abnormalities that are seen in Parkinson's disease, such as tremors, slowed movement, impaired speech, or muscle stiffness. Okay, so in terms of alterations, I had already told you that we have loss of dopamine neurons, which have an inhibitory effect, so less dopamine ends up being produced, and that the areas affected are the substantia nigra here, as well as the basal ganglia collectively here. So a little bit more on Parkinson's disease. I already told you that there's dysfunction in the neurotransmitters that are produced within the brain. We talked about dopamine. On this slide, we're going to talk about both dopamine and acetylcholine and how they have an impact on the development of Parkinson's disease. The cause of Parkinson's disease can be oxidative stress, mitochondrial dysfunction, loss of nerve growth factors, but it can also be genetic predisposition. With a genetic predisposition, we end up with accumulation of a protein called alpha-synuclein, which you can see right here. And what happens is this protein misfolds and becomes non-functional. And the protein accumulates and then ends up forming Lewy bodies, which are concentrated within the basal ganglia. And it ends up causing degeneration of these dopamine-containing neurons that terminate here, that arise from the substantia nigra. Okay, so that's written here. We get the neurodegeneration occurring, reducing or losing that production of dopamine. And then we end up with a dopamine deficiency, and we can end up with loss of normal inhibition, which we'll talk about shortly, or normal inhibition of muscle tone. So before I continue on down here, a couple of things I wanted to go over. First of all, a nerve that produces dopamine is called a dopaminergic neuron, and a nerve that produces acetylcholine is called a cholinergic neuron. What I'd like to do now is spend a few minutes talking about the relationship between acetylcholine and dopamine in normal individuals. Oops, so let's move this over. So dopamine is produced in cell bodies within the substantia nigra, as I already spoke to you about. And these are dopaminergic neurons then, as I already told you. And they release dopamine within the striatum. Acetylcholine at this point is then produced by nerves within the striatum. So we have both dopamine that is released here within the striatum, which has an inhibitory effect. And then we have acetylcholine that's produced by cholinergic nerves or neurons within the striatum. And acetylcholine is then also released here. And acetylcholine has an excitatory effect. Okay, so you can indicate up here that it is dopamine, DA for short, that has the inhibitory effect, and acetylcholine that has the excitatory effect. So it's considered an excitatory neurotransmitter. So again, both dopamine and acetylcholine are released here within this neuron. And you can think of this concept similar to a car where you have an accelerator pedal and you have a brake pedal. And between the two, you can regulate the speed of your car. And so you can see that here, with proper functioning, we have a nice balance of dopamine and acetylcholine. So we have that balance between the inhibitory effect, or the braker, and the acetylcholine effect, or the accelerator. So this balance is what leads to controlled movement. Now, in terms of those with Parkinson's disease, you can see here that we have an imbalance between dopamine, our inhibitory neurotransmitter, and acetylcholine, our excitatory neurotransmitter, because of the destruction of the neurons. And because of this imbalance, we end up, as you can see in this light green box here, having unopposed activity of the cholinergic neurons, or the acetylcholine neurotransmitter. And what this is going to do is continue to promote muscle tone, which ends up leading to some of the symptoms that are common in Parkinson's disease, such as resting tremors, rigidity, bradykinesia and akinesia. Both of these terms and a couple of other terms are listed on your PowerPoint for your own reading. And so the final result then, and compared to having the nice controlled movement as we would with a normal person without Parkinson's disease, we end up with very disturbed, uncontrolled movement because of the loss of that inhibitory neurotransmitter dopamine and the uncontrolled or unopposed effect of acetylcholine. Now, the third condition that we will speak of is called Huntington's disease, which is also called Huntington's chorea. And it is an autosomal dominant trait with high penetrance. Remember that autosomal dominant is one of the several ways that a trait or disorder can be passed down through families. So if a disease is considered autosomal dominant, it means that you only need to get the abnormal gene from one parent in order for you to end up inheriting the disease. And just another recall that penetrance is the extent to which a particular gene or, say, set of genes is expressed within the phenotype of individuals carrying it. Children of Huntington's disease have a 50% chance. Children of parents with Huntington's disease have a 50% chance of actually getting the disease. With Huntington's, the problem is on chromosome 4, where there is a higher than normal CAG trinucleotide repeat on specifically chromosome 4. So normal is anywhere from 9 to 34 repeats of the CAG trinucleotide. But in Huntington's disease, we have anywhere from 40 to 70 of these repeats. And the higher number that you see, generally the sooner the disease will show, so the earlier the onset, and also the more severe the condition will be. In addition to that, people that have say high 30s in terms of repeats may have the possibility of developing Huntington's disease. Age of onset is anywhere between the ages of 25 and 45 years of age, and unfortunately the trait at this point may have already been passed onto their offspring. So you may have had children and then you end up showing the symptoms of Huntington's disease after you've already passed on the trait potentially to your children. So that's obviously a concerning component if you didn't have genetic testing or you weren't aware that Huntington's may have run in your family. And the underlying changes that are seen in the central nervous system is the accumulation of tangles of the abnormal protein, Huntington protein. And it's abnormal because we don't normally possess this protein within our body. And over time, this accumulation of this Huntington protein, which of course is abnormal, will end up killing off the cell. So in the case of Huntington's disease, it is the GABA-producing neurons that are affected. GABA is an inhibitory neurotransmitter that acts like a brake and inhibits action potential firing. So what it will do is it will actually, if our resting potential is running along like this, what GABA will do is drop the resting potential, making it more difficult to achieve an action potential. So when GABA binds to a receptor, one of the effects that GABA has is it's going to open up a chloride channel that is going to allow chloride ions to enter the cell. And when we change the what ions are present within the cell, it ends up changing or reducing the membrane's potential, making it, again, more difficult to reach an action potential. So these GABA neurons, or GABA-producing neurons, they're found within the basal ganglia, and they are also found within the cerebral cortex. So the basal ganglia and the cerebral cortex possess these GABA-producing neurons. And we end up, because of the loss of this inhibitory effect from GABA, we end up with affecting the effect of a loss of voluntary movement control. again, because this is due to the alterations within the basal ganglia for voluntary movement, but we also see issues with mental function or cognitive deficits because of alterations that are seen within the cerebral cortex and the death of the neurons in that area. And specifically, what ends up causing the loss of these inhibitory GABA neuron or GABA producing neurons is that chains of glutamine are going to end up sticking to each other. So these chains are found on this abnormal Huntington protein. They end up sticking to each other, and over time with accumulation they end up contributing to the loss or death of these neurons. With people that have Huntington's disease and it's starting to progress, they'll end up with progressive dementia, progressive cognitive deficits, so loss of their working memory, reduced capacity to plan and organize and sequence activities, slow thinking, apathy, restlessness, disinhibition, irritability. And what's a key feature of Huntington's disease are the involuntary movements referred to as chorea, athetosis, and ballism, all of which I've included the definition of within the notes of your PowerPoint. And on a few of the slides, you'll see that there is a YouTube video link. You can, if you'd like, click on that just to see a short blurb on the disease itself. The next condition is multiple sclerosis. Multiple sclerosis is an acquired autoimmune inflammatory disorder. It can be triggered by such events as trauma, pregnancy, possibly viral infections. There's a possible genetic component to it. Only 15%, though, of people with MS have an affected relative. So the genetic component is still sort of uncertain. And it affects women greater than men. Age of onset is anywhere between the ages of 20 and 40 years of age. And it can go through relapses and remits. So the person ends up going through a cycle where they're getting worse, and then they stabilize, then they're getting worse, and then they stabilize again. Lots of ups and downs. And these relapses and remissions, they can go on for about 20 years, and then at that point, it will often end up in some type of disability. On the diagram here on the right, you can see that this is a normal nerve where the cell body is going to be receiving signals from the dendrites, and then the signal sent down the axon until it reaches the axon terminal, where it would release its neurotransmitter communicating with whatever it's synapsing with. And you can also see the presence here of myelin. And in MS, it is the myelin sheath that is damaged. In particular, though, it's actually damage or destruction of the oligodendrocytes, which actually produce myelin within our central nervous system. And this ends up leading to the formation of plaques or scarring or lesions. But the axons themselves are preserved. So this disease is attacking, it's an autoimmune disease that's attacking the myelin sheath by causing destruction of the oligodendrocytes, which form the myelin sheath, but leaving the axon itself spared. So when we have loss of myelin, because we know that, if you remember, myelin is involved in speeding the conduction. And so if we have loss of myelin, we end up with reduction in conduction speed, leading to overall loss of function. And some of the effects that are seen are paresthesia, so tingling, burning, tickling sensation on the face, trunk, or limbs. Weakness is oftentimes seen, visual disturbances, as well as possibly urinary incontinence, depending on the person that is affected. And so if the myelin sheath is lost, then the ability to have an action potential actually move along the axon as it should, continuing to be able to pick up speed and travel where it needs to go, ends up being diminished or lost. The next condition we'll talk about is amyotrophic lateral sclerosis, also known as ALS or Lou Gehrig's disease. Just to break up the term here, oftentimes in front of a term refers to lack of, myo referring to muscle, trophic referring to growth, and lateral referring to the sides, and sclerosis referring to plaque formation. The cause of ALS is really unknown. It's possible or it's suspected that maybe there's a genetic component. Men are more likely than women to be affected by ALS, and age of onset is commonly between the ages of 40 and 50. But what's very sad is that death occurs usually within 2 to 5 years after onset. And the reason for this is because of the loss, usually because of the loss of respiratory muscles, but I'll get back to that shortly. So ALS affects the peripheral muscles and ends up decreasing the growth of the muscles, causing overall degenerative changes. And the disease in particular affects motor neurons. So it affects upper motor neurons and lower motor neurons. Your upper motor neurons are found mainly in the brain and part of the spinal cord, and your lower motor neurons are found more within the spinal cord. And what's interesting is that even though we have degeneration of these motor neurons, it is without inflammation. And of course, the areas affected then are the brain and the spinal cord. This is the initial areas that are affected because, of course, everything that would need to be supplied by these areas ends up being affected too. And so if you look at this diagram here, we have a normal, healthy-looking neuron supplying a muscle, but in ALS, we get degeneration of that neuron and subsequent degeneration and loss of function of the muscle that it supplies. So ALS affects voluntary motor functions or control of voluntary motor functions. So it affects skeletal muscle, for instance, and that's where it gets us back to the respiratory muscles, and it affects those respiratory muscles that we contract and we have control over in terms of our breathing. And when we lose control over these muscles, that's usually when death occurs, because we're not able to complete one of life's required functions, which is that of respiration. Some of the effects include progressive muscle weakness. So it tends to start in the arms and the legs, which is where the term lateral comes from, or when we spoke about the periphery, and then it just continues to atrophy. We'll end up seeing spasticity as well as loss of manual dexterity and gait, so having a very difficult time controlling those voluntary muscle functions that to the average person, you don't even have to think about it. Now we'll talk for a few minutes about the cause and mechanism of destruction that is involved in the disease ALS. So the specific cause has to do with glutamate, which you should make note of as being an excitatory neurotransmitter in the brain, which in normal healthy individuals, glutamate reuptake occurs by glial cells, but in those with ALS, this function is reduced, and it ends up leading to the accumulation of glutamate, which, when accumulating, ends up having an excitotoxin effect, leading to neurotoxicity. Glutamate release is normal, as I told you, and this reuptake then ends up changing or reducing, and then we end up with subsequent toxicity, and this toxicity kills the neurons that are found within both the upper and neurons of both the upper and lower motor neurons. In the upper motor neurons, so the brain and into the spinal cord, we end up with axon degeneration, demyelination, sclerosis or scarring. In the lower motor neurons, we end up seeing that the motor unit itself is denervated, and this ends up leading to muscle atrophy and weakness, because obviously the skeletal muscles are not getting the nerve supply that they need to function. So what ends up happening really is that overall communication ends up being initially diminished but eventually lost. So what we spoke about on the previous slides were disorders that are commonly, the common age of onset is in adulthood. But now on the next 3 slides, we'll speak about a few different conditions that are more so found in, or commonly found, not commonly, but found in children compared to adults. And the first one is a metabolic derangement called phenylketonuria, or PKU for short. And there's a small amount of infants that don't possess the enzyme called phenylalanine hydroxylase. And what phenylalanine hydroxylase does is it breaks down phenylalanine into tyrosine. And this would just be a normal process that happens all the time within our body, but some infants are born without this enzyme. And without this enzyme, we end up with a buildup of phenylalanine that accumulates to toxic levels. But we also end up without the production of tyrosine. And both end up having an effect. So looking at the image here, so phenylalanine is a component of a normal diet. So it's found in a lot of different foods, foods that are higher in protein, for instance. And when it is not broken down into tyrosine, then it ends up with this decreased tyrosine, we end up with a reduction in serotonin levels. And serotonin is involved in mood control, so depression, for instance. And it also reduces the levels of catecholamines within our body. Catecholamines, examples would be norepinephrine, epinephrine, and dopamine. And it also ends up leading to decreased melanin. And melanin is a pigment that's found within the skin, the eyes, the hair, giving color. So people that have PKU can end up being very fair-skinned, have very light eyes, and very light hair as well. So these are the 3 main effects: the reduction in serotonin, the reduction in catecholamines or the decreased dopa, as well as decreased melanin, all because of the reduced formation of tyrosine because of lacking the enzyme that breaks down phenylalanine into tyrosine. But there's also the effect of increased levels of this phenylalanine within the body. And what it can do is damage the central nervous system, so it has more of the accumulation of phenylalanine has a much greater effect. And it can lead to issues such as mental retardation, seizures, hyperactivity, if it's not dealt with early. So if somebody is born and they're diagnosed with PKU, it's important that it's dealt with appropriately early, and it can be detected right at birth. And how it's dealt with mainly in terms of the buildup of phenylalanine is that the individual has to have a diet that is very low in foods that in what would contain phenylalanine. And how this condition is detected is right at birth through a heel stick test. The infant is pricked at the heel and samples are taken, and tests are run to see if the enzyme is or is not present. And then the last thing I wanted to mention on this slide is that the importance of tyrosine we already discussed, but tyrosine is used in our body to make proteins, to make melanin, to make thyroxine, to make norepinephrine, dopamine, as well as 5-hydroxytryptophan. And when we struggle with producing these, we end up with deficiencies in other areas. The next disorder that we'll speak of is called Spina bifida, which is a type of defect that happens within the neural tube. So this happens when a baby is developing in utero. And if we look at the diagram here on the right, this is a normal spine, and you can see that the spinal cord is nicely contained along with the meninges nicely contained within the vertebral column. Then there's something called a meningocele, which is less serious than the next one that we'll speak about. And with a meningocele, you can see that what happened in utero is that the meninges have protruded outward, like pushing outward, and you can see the level of the skin around it, and it's filled with cerebral spinal fluid. And that's really all that this outpouching contains. But it's important that it's fixed, because if it isn't, then over time, the spinal cord itself can herniate into this area. And as you can imagine, as the child grows, the impact of the stretching in this area, what that could do and how it can predispose that spinal column to herniating. But what's important to know about a meningocele is that there aren't any neurological deficits, because the spinal cord and the nerves are left intact. Okay, so that's a meningocele. And then we have spina bifida, or myelomeningocele. And the biggest difference here is, yes, we have that outpouching of the meninges, but we also have the spinal cord outpouching or within this area as well. Nerves are also found, and of course cerebral spinal fluid. And as this usually happens in the lumbar or sacral region, but as you can imagine, when this person grows, the spinal cord ends up being stretched within this area, and it can lead to scarring as well as various deficits that would be found below, maybe at or below the level of the lesion. And so early intervention is, of course, very important. Fortunately, this is something that if somebody does have it, it's identified when a mother goes to have her anatomy ultrasound. One of the things that the technician is looking for is the presence of any neural tube defects, and this, of course, is a big one. So it can be identified very early, and then you can intervene very early as well. But as you can imagine, the outcome is not as good for people that have spina bifida myelomeningocele compared to people that have just the meningocele. Another thing I wanted to mention here is that in people that have spina bifida, 85% of these patients will also have hydrocephalus. And if you remember hydrocephalus, which we'll actually talk about on the next slide, the water or excess CSF found on the brain. And then the last thing I should mention is that I spoke to you about the scarring and some of the deficits that can occur at or below the level of the lesion. But as you can imagine, you would end up with abnormalities in your gait, as well as both bladder and sphincter dysfunction as being, some of the major issues that are seen with this condition, especially if it's left untreated. And then the last condition we'll talk about is congenital hydrocephalus. Congenital refers to being born with, so this would be different than acquired hydrocephalus, which might come on, say, after certain injuries where CSF production increases, certain conditions that can come on later on in life and be acquired. We're talking about congenital, so something a child is born with. And what hydrocephalus, hydro referring to water, but specifically referring to the presence of CSF, and cephalus referring to the brain, or on the brain. So you can see here a normal brain of an infant, and you can see that of a child that has hydrocephalus. And the CSF that's normally found within the ventricles and is overproduced and fills the ventricles. The ventricles end up enlarging, as you can see here, and when they enlarge, they push the brain up against the skull. Now, because the baby has a more their skull is more flexible because of the presence of if you remember fontanelles, they don't have the fusion of the sutures like adults have, and so when it protrudes against the brain, the head actually gets larger because it can sort of accommodate the space or the pressure, and you can even get bulging out that fontanelle, so that's the soft spot on a baby. And what this can lead to, especially if it's untreated, is a cranial deformity. But it can be treated with, in many cases, with shunting. So they put a shunt in that allows proper draining of this cerebral spinal fluid back into the venous system. Just back up at the top here to speak about the congenital causes. There's different causes. The baby could be overproducing CSF. There could be an obstruction of the normal resorption of CSF, or just entirely lacking the ability to resorb the cerebral spinal fluid. And so then it remains in the ventricles and puts a substantial amount of pressure on surrounding brain structures. And so the timing of hydrocephalus, I already spoke to you about, whether or not it occurs before fusion of cranial structures or after fusion of cranial sutures, I should say. And children will have fusion of where the fontanelle closes, and their sutures will form and fuse. It happens at different ages. So sometimes it happens at the age of 2, or it can be a little bit earlier or a little bit later. So timing is obviously very important, because if the cerebral spinal fluid continues to increase after the sutures have fused, then we get increased brain volume pushing against fused skull bones, and there isn't any accommodation or flex in this area. And then here's just an example of the normal ventricles, and then ventricles filled with excess cerebral spinal fluid, and you can just see how much larger the area is that's filled with cerebral spinal fluid, how much more expanded these ventricles are in normal versus a brain that is experiencing hydrocephalus. And then over time, if it's untreated, and this brain volume continues to put pressure against the bones, then it can cause neurological deficits, but also inhibit the central nervous system growth of that infant or child. So that is the end of the second lecture on neurological disorders. Thanks for listening.